Dear students, அனைவருக்கும் வணக்கம், வந்தனம், நமஸ்காரம். Dr.N.Sathyamoorthy from Trichy. Today I'd like to discuss 2 and 3 more questions in biotechnology. Biotechnology is the 4th chapter in botany. Applying technology in biology is said to be biotechnology. Genetic engineering, plant tissue culture, and protoplasmic fusion, or the branches of this biotechnology. I like to discuss only the 2 and 3 more questions today. Are you ready? Question number 1 for you. What are the materials used to grow microorganisms like spirulina? Microbe spirulina, it's an example for blue-green algae, and it is used as a single-cell protein. Spirulina-like microorganisms, the cyanobacteria, are easily growing in fresh water because it's a freshwater algae. It's an autotroph. It has got the ability to synthesize its own food material. And we can grow this spirulina artificially in aquarium tank and photobioreactor. Fermenter or the bioreactor, helps to produce large-scale biomass. Photobioreactor means it's a photosynthetic cyanobacteria, the blue-green algae. So if light is present, it has got the ability to synthesize its own food material. So we grow spirulina in fresh water, artificial aquarium tank, and in photobioreactor. Other unicellular microorganisms used as protein diet for human consumption or fodder for animal, that is SCP, are grown in paddy straw, sugar waste molasses, animal manure like cow dung, and the sewage. So we grow microorganism in freshwater or aquarium tank or in photobioreactor, and other unicellular organism, especially the saprophytic organisms, in paddy straw, sugar molasses, animal manure, and the sewage. Okay. Then the next question for you, the second question. Name the chemicals used in gene transfer. Gene transfer means transferring the DNA fragment from one organism to the other. It's an important criteria in genetic engineering. Gene transfer means transferring the DNA from the donor to the host. Or through a vector, we are transferring the gene to the host. 2 chemicals are used. One is polyethylene glycol and another is the dextran sulfate. These 2 chemicals used in gene transfer at the time of genetic engineering. Polyethylene glycol, it also used as a fusogenic agent at the time of protoplasmic fusion. Protoplasmic fusion, fusing the protoplast of 2 similar species or 2 different species or 2 different genera to produce a hybrid cell. So this polyethylene glycol acts as a fusogenic agent in protoplasmic fusion and also helps in gene transformation both in bacteria and the yeast. Dextran sulfate, also used in gene transfer, it's a very good anticoagulant. Coagulation means clumping of cells, So it prevents clumping of cells at the time of gene transfer. Okay. Next question for you, question number 3. How do you use the biotechnology in modern practices? Biotechnology is a newer technology. The technology is implemented in biology, botany, zoology. Biotechnology used, that is the uses of biotechnology, simple as it's used in enzyme and biomass production. So large scale production of enzymes, we use fermenter, and also for biomass production for SCP, single cell protein. It is used to produce secondary metabolites, antibiotics, and vitamins. Metabolites. Metabolites are the end products of metabolism. The network of biochemical reactions occur in all normal living cells is said to be metabolism. And this metabolism is of two types. One is anabolism, another is catabolism. Anabolism means biosynthesis, synthesis of macromolecules from its monomeric unit. Photosynthesis is a very good example for anabolism. Breakdown of macromolecules into their monomeric unit is said to be catabolism. Respiration is an example for catabolism. This metabolism produces primary metabolites and secondary metabolites. Primary metabolites are the chemicals produced in large quantities. Example, carbohydrates, fats, proteins, or nucleic acid, like that. And secondary metabolites are produced by the plant in very minute quantities. It is very useful to other organisms, not to the plant. And antibiotics. Antibiotics are the chemicals or chemotherapeutic agents isolated from one living organism and used to kill the other. Penicillin, streptomycin, aureomycin, neomycin, bacitracin, subdilin. These are all produced in large scale by using the fermenter, the bioreactor. And vitamins, fat soluble and water soluble vitamins, we can produce vitamins also through these biotechnology, especially large scale production. Then it is used in biofuel production. Biofuel production using algae. Algae, especially the blue-green algae, has the ability to produce, cyanobacteria has got the ability to produce biofuel. So large-scale production of biofuel and large-scale production of biofertilizers. Fertilizers of biological origin are known as biofertilizers. Acetobacter, Clostridium, Rhizobium, like that, we can produce in large quantities. And used in recombinant DNA technology. By using our DNA technology, we are producing insulin, human insulin in the fermenter. That insulin is said to be humalin used to treat diabetes. So all these things are the uses of biotechnology to us. Next question for you. I'm just learning technology. That's why the problem. You are working in a biotechnology lab are working in a biotechnology lab and using the bacterium, intestinal bacterium, E. coli. E. coli is an example for commensals. In an association of two living organisms, one is benefited and the other is not affected. We are calling it as commensal. This E. coli is the intestinal bacterium. How can you cut the nucleotide sequence? Explain it. So if you want to cut the nucleotide sequence, you use the restriction enzyme or the restriction endonucleases isolated from the bacteria. So with the help of restriction enzyme, I cut the nucleotide sequence. Okay, in biotechnology, this restriction enzyme used as a molecular scissor. And the restriction enzymes are isolated from the bacteria. The main function of restriction enzyme is to cut the invading viral DNA into pieces. So it's a kind of protection seen in bacterium to or kill the viruses, the bacteriophages. Viruses infecting the bacteria are known as the bacteriophages. So we can cut the nucleotide sequence or DNA using the restriction enzymes. Then I hope types of restriction enzymes, if you write, it is useful. There are three classes of restriction endonucleases, exonucleases and endonucleases. Restriction endonucleases, it can cut the DNA in the middle of the strand. Three types of restriction endonucleases are reported, namely type 1, type 2, and type 3. In our DNA technology, recombinant DNA technology, in genetic engineering, generally we use type 2 enzymes. This is very important. One more question also. Okay. Then the specialty என்ன அப்படின்னு கொடுத்திருக்கோம். What is the specialty of this enzyme? This enzymes recognize and cut the DNA in a specific nucleotide sequence. So particular area, it has got the ability to cut. For example, if you take the endonucleases, see this is a DNA strand, DNA duplex. in this DNA duplex, a restriction endonucleases cut the DNA in the middle. Some restriction enzymes create even cuts. Some restriction enzymes create uneven cut in the DNA strand. Okay. So this is even cut. This is uneven cut. This is a structure of restriction endonuclease or restriction enzyme. And the restriction enzyme has got the ability to identify 4 to 8 base pairs. Adenine, guanine, thymine, and cytosine, or the nitrogenous base pairs are seen in DNA. Adenine, guanine, cytosine, instead of thymine, uracil is present in RNA, Then what are the enzymes you can use to cut the terminal end and internal phosphodiester bond of nucleotide sequence? This question helps to explain the types of restriction enzymes. Okay, what are the enzymes you can use to cut terminal end of the DNA sequence and the internal middle of the DNA phosphodiester bond of nucleotide sequence? Nucleotide is the nanoparticle of life, and nucleotide is a combination of nitrogenous bases, sugar, and phosphate. Nucleotide is a combination of, 1 minute. Nucleotide is a combination of nitrogenous base. Adenine is always pairs with thymine with the help of 2 hydrogen bonds. This nitrogenous bases combines with sugar to form a nucleoside. So here thymine also combines with the sugar to form a nucleoside. This nucleoside combines with a phosphate molecule to form a nucleotide. Okay, this is a nucleotide, the nanoparticle of life in human genome. 6.6 into 10 power 9 nucleotides are present. This restriction enzyme has got the ability to cut the terminal end of DNA as well as the internal phosphodiester bond. How? I already said that 2 kinds of restriction enzymes are reputed. One is the exonucleases, another is endonucleases. See here, this is exonuclease enzyme. Exonuclease enzyme are used to cut the terminal end of nucleoside. So nucleotide oda terminal end irukku pathilona adha cut pannum terminal end. Adha inga kuduthirukku. Example for this exonucleases ball 31 and exonucleases 3 example for exonuclease. Then we go to the endonucleases, the next page, wait for a while. Then the endonucleases. See here, endonucleases. Endonuclease enzyme are used to cut the internal phosphodiester bond of nucleotide sequence. The HINDA 2 and ECOR1 are the best examples. See this picture here. Some restriction enzyme creates even cuts in 2 strands because DNA is made up of 2 helices or 2 strands. Some restriction enzyme has got the ability to cut the 2 strands in the same position. We are calling it as even cut, leads to the production of blend ends. But some restriction enzymes create uneven cut. See here, first cut here, in the opposite strand, the cut is here. So it creates uneven, this cut is called uneven cut, and it creates sticky ends. So restriction enzyme creates even as well as uneven cut in the DNA strand. Okay. Next question for you. What do you know about the word PBR322? PBR322. It's a plasmid. Plasmid is a non-genomic DNA or extracircular DNA present in the cytoplasm of the bacterial species. It's artificially or lab-made plasmid, PBR322. It's a reconstructed plasmid in the lab. It is widely used as cloning vector in genetic engineering. Somebody else carry the gene from the donor to host. We are calling it as vector. In zoology, you know the vector-borne diseases: malaria, filariasis, Japan's encephalitis and dengue, chikungunya, all these diseases are vector-borne diseases, the pathogen carried by one organism to the other with the help of a vector. Vector na carrier, Anopheles mosquito, female Anopheles mosquito, female Culex mosquito, female Culizin mosquito, and female Aedes mosquito act as a vector to carry the pathogen from one to another organism. So this this plasmid as well as bacteriophages in animal system, retroviruses act as a vector to carry the gene from the donor to the host. This PBR 322 plasmid are composed of 4,361 base pairs, ATGC. And in PBR, the P denotes the plasmid, B and R denotes the scientist who reconstructed the plasmid. The B represents the scientist Bolivar and R represents the scientist Rodriguez. So these two scientists developed a PBR322 plasmid. Okay. And I will show the structure of this PBR plasmid also. And the number 322 represent the number of plasmid developed from their laboratory. So they gave a number 1, 2 like that. This is a 22nd plasmid developed in the lab. And this plasmid possesses antibiotic resistant gene or the marker gene, ampicillin resistant and tetracycline resistant. Two different antibiotic resistant genes are present. See here, ampicillin and tetracycline. And it also possesses a lot of recognition site by the restriction enzyme or restriction endonucleases. We are calling it as restriction enzyme sites. So these sites are identified by the restriction enzyme and it cut DNA on that region. And this plasmid also possesses origin of replication. This is very important. 1 minute. Okay, it possesses origin of replication. This is very important one, where we can add the foreign gene. And how will you identify vectors? Vector means a carrier. How will you identify a vector, or what are the characteristic features of vector? The vector should be smaller in size. And first, in biotechnology, we are using plasmid, bacteriophages, and retroviruses as vectors. This vector should be a smaller one, and the molecular weight is less than 10 kilobases. Kilobases means 1,000 base pairs. 10 kilobases means it will be around 10,000 base pairs. It can indefinitely replicate within the host cell. It has got the ability to replicate, self-replication ability. Generally, the vectors possesses this ability. The replication, self-replication, plasmid is a self-replicating structure. And it should contain suitable marker for antibiotic resistance. Now, it may have either ampicillin resistant gene or tetracycline resistant gene we use as a marker. And vector must contain origin of replication. We are calling it as ORI region. It should possess as ORI. And cloning vectors have more than one restriction sites. So we have a lot of more than 900 restriction enzymes isolated from the bacterial species. So this plasmid or bacteriophage or the retroviruses should have more than one restriction sites. Then we can use it as a vector. Next question for you. What are the advantages of herbicide-tolerant crops? Herbicide or the chemicals used to kill the herbs are called herbicides, also known as weedicides. Right, the advantages of herbicide-tolerant crops means the plant has got the ability to withstand the herbicide used in the field. This herbicide-tolerant crops improves the crop yield because it is tolerant to herbicides, so nothing will happen, no harm to the host by the herbicides. It reduces the use of herbicides. Already it is herbicide tolerant, so we shouldn't use herbicide in the field. Then improve the soil fertility. Lesser use of herbicide helps to increase the soil fertility, soil microflora. Reduce the environmental toxicity. Herbicides or the chemicals naturally affect the environment. Lesser use protect the environment from the herbicide toxicity. It conserves the soil living microbes, especially bacteria and fungi, the decomposers. Herbicide tolerant crops we are cultivating means we use or not use the herbicides. So it helps to enhance, helps to improve the growth of microbes present in the soil, especially soil fungi and soil bacteria, the decomposers. Decomposers of the organism has got the ability to decompose the dead organic matter and convert the organic substances into inorganic substances, especially minerals, helps to improve soil fertility. Okay. Then next question for you. Compare the various types of blotting techniques. Blotting, three kinds of blotting described in your book. One is southern blotting, another is northern blotting, another is western blotting. Okay, wait for a while. Dear students, what is blotting? So blotting is the process of transferring macromolecules like proteins or nucleic acids, DNA or RNA, from a gel to the solid surface of an immobilized membrane to detect the molecule that have been transferred. So blotting is nothing but the process of transferring macromolecules like proteins or the nucleic acids like DNA or RNA from a gel means agar gel, to the solid surface of an immobilized membrane. It may be nitrocellulose or amino benzyl oxymethyl or nitrocellulose to detect the molecule that have been transferred. So this blotting, three kinds of blotting reported in your book. One is the southern blotting. Then second is the northern blotting, and third is the western blotting. So what are the characteristic features of these three blotting? First is southern blotting. It is the name of an inventor who invented this technology. And northern blotting is a misnomer. It is not the name. And western blotting is also a misnomer. This southern blotting helps to separate DNA. Northern blotting helps to separate RNA. And western blotting helps to separate proteins. And for this southern blotting, denaturation is needed. For northern blotting, denaturation is not needed. But for western blotting, denaturation is needed. What are the membranes used? For southern blotting, we are using nitrocellulose. For northern blotting, we are using amino benzyl oxymethyl. And for western blotting, we are using nitrocellulose. Hybridization here, DNA to DNA, here RNA to DNA, here protein to antibody. Then how will we visualize this blotting? Southern blotting visualized by using autoradiogram, and northern blotting using autoradiogram, and western blotting we use darkroom. So the three types of blotting described in your book, and the table is also given in the book. So thank you very much for hearing. Next class I will explain the important 5-mark question asked in your textbook. Thank you very much. Thank you. Thank you. Thank you.